In this lecture, I'm going to combine chapters 10 and 11. And the reason why is because I think that these two chapters fit together really nicely. And the underpinning or the underlying theme in both of these chapters is going to be an emphasis on time. Now, I know when we're looking at speciation, that the first thing that doesn't necessarily pop into our head is time. But really, when we talk about speciation, and we talk about individual species, the thing that we have to focus on is we have to focus in on that concept of time. So what I am showing you here with this first lecture slide is that we have a phylogenetic tree, and this phylogenetic tree is mostly for the mammals. I like this phylogenetic tree. It's a slightly different orientation than what we have seen before, and we can see that connection to the Cambrian explosion that we'll talk about a little bit later. And then we can also see those major mass extinction events. And that's, of course, going to be important for how that contributes to or how that affects the later species that come about. So just a quick reminder about speciation. Remember that speciation is the splitting of one species into two or more species, and that we're really dealing with gene pool changes. We're dealing with those changes in the DNA, whether that's changes in just one gene that causes the new species to be unable to breed with the old species or the original species, or if we're dealing with lots of accumulated changes, or if we're dealing with all of the above that is driving that speciation. And our example here, where we see significant differences, is in the American robin and the eastern bluebird. They often live in the same geographic location. They both consume a significant amount of insects, and yet when we look morphologically, they are very different from one another. Sometimes when speciation happens, like with the flycatchers below, the changes are very, very subtle, and it's hard to see the differences between those species. So, in this chapter, we look at some of those mechanisms that are driving speciation. Prezygotic isolating mechanisms are all the things that happen before the zygote is made. So we're talking about the sperm and the egg coming together, and what are the things that are happening before the offspring are made. So some of the prezygotic isolating mechanisms will be habitat, temporal, behavioral, and mechanical and gamete. So when we look at habitat isolation as a prezygotic isolating mechanism, what we see is that organisms or species will often have different places where they like to breed. So again, here's our example of flycatchers, and we see the two maps where they prefer to breed. And what this means is that the habitat preference is what's keeping these species separated from one another so that the sperm and the egg could never even combine in the first place. Another example that we see is with human intervention, human fragmenting of habitat. We are then stepping in and preventing the sperm and egg of populations to combine, or the sperm and egg of separate species to combine. Now, it's not our intention, but it is often the result. Temporal isolation has to deal with time, and this is species that are separated by the timing of their breeding. So in our area, we have the American toad and the Fowler's toad, and based on when they want to breed, that is what's going to keep their sperm and their egg separate. Behavioral isolation has everything to do with male and female, primarily, behaviors that they're looking for in order to identify if the species or the individual is a male and a female and whether that breeding should occur. So this often happens when we have species that are breeding at the same time. And so all the males and all the females of many different species are together. They're all wanting to mate, and the hormones are very high, so mistakes could happen if there wasn't this other level of looking for specific behaviors. And that's where we get this example of our fireflies. Mechanical isolation, I think, is the most straightforward. It really is all about, can you physically get the structures that hold the gametes? Can you get those physical structures together? Can you exchange your gametes? And our example here of the snails, has to do with which way they make their shells. So if you make your shells in opposite directions of one another, no matter how much you may want to get your gametes together, you physically cannot exchange your gametes. Now with gamete isolation, this is really for species that are releasing their sperm and their egg into the environment. And so there is no other control mechanism. The cells are separate from the parental body. And so the parental has no control. And so what's happening at that cellular level? How are the species able to control fertilization and to make sure that their sperm and their egg combine that are of the same species, and that the sperm and the egg aren't combining of separate species and potentially leading to some issues? And so one of the things that can happen is the egg may secrete a chemical to stop the sperm from being able to enter if the sperm is not of the same species. Now in plants, when the pollen is floating through the air, the pollen can land on any female structure, and on the female structure are chemicals that are stopping the pollen from germinating or from growing, so stopping fertilization from happening unless that pollen is of the same species. So those are some of the prezygotic isolating mechanisms that we see for keeping species separate. But what if the sperm and the egg combine anyways? What if you've made that zygote? Are there any isolating mechanisms for what could happen if sperm and egg combine of separate species? So one of the things that will happen is zygote mortality. Pretty self-explanatory. Sperm and egg came together, but then the zygote, the baby, the offspring, did not survive. Then we have hybrid sterility and F2 fitness. Now with hybrid sterility, this is where the sperm and the egg of two separate species, say a donkey and a horse, come together, and they make offspring that are healthy enough. But if those offspring go to make their own offspring, they cannot. So when a donkey and a horse come together to make a mule, the mule can have a perfectly happy little mule life, but the problem is that mules cannot mate with each other and make more mules, which is why we don't see mules as a separate species. You have to keep remaking mules from mating donkeys and horses together. Another example of post-zygotic isolating mechanisms will be F2 fitness. So we can see this happen with horses and zebras. And with horses and zebras, there are enough genetic similarities that they can make zorses. And zorses can actually mate together and make more zorses. But the problem is that the zorse babies are all going to have decreased fitness. They're not very likely to reach reproductive maturity, which is why we don't see a zorse as an entirely separate species. So the parents can come together and make babies, but when the grandbabies are made, that F2 generation, that's where we see a lot of the issues and the problems show up. So here we have three very common isolating mechanisms for if the sperm and the egg of separate species have combined. So what we see is species integrity. Most zebras look the same. Most horses look the same. We see this consistency and continuity within a species, and we see that oftentimes over multiple generations. So remember our two models of speciation, the gradualistic model and the punctuated equilibrium model. Now, the gradualistic model would say that species are gradually changing over time, and that we often don't find the last most recent common ancestor living alongside the existing newer species. And then our punctuated equilibrium model says that species actually stay the same for a long period of time. We have that consistency and that continuity over multiple generations, but then a sudden speciation event occurs, and we now can say that that's due to genetic changes, whether lots of changes in genes or just a change in one important gene, and then we finally have speciation that occurs. So remember, the punctuated equilibrium model is what is supported by research and scientists today, and that the gradualistic model was our historical way that scientists and researchers and naturalists were trying to explain observations like zebras and horses, the continuity, but then also the idea that you could get things like a zorse. Now, The one thing that we have to come back to again is time. So time becomes important when we happen to step in or catch speciation in the process of happening. So one of the things that can happen is we can have these things called hybrid zones. So on the left hand side here, we see the little blue dots, and all those blue dots are representing individuals within a population. And we see that there's lots of gene flow. The breeding individuals are breeding with each other and consistently producing generation after generation, organisms that look very similar and have shared genes. A lot of similarities in the gene pool. But if we get a barrier, whatever that barrier may be, and we separate the populations, what we have done is we separated the gene flow. So our population down here at the bottom that stays blue, we see that within that population there's still a lot of gene flow, a lot of breeding with the individuals. We see consistency and continuity in the phenotypes from generation to generation. But in the top arrow, where our little dots start to change from blue to pink, what's happening is we see a change in genes, either the actual genes themselves or the gene expression. We're starting to see some phenotype changes. But for a while those changes are happening from one generation to the next, there are still enough genetic similarities that should an individual from the pink population up on top make it down to the population in the bottom arrow with all the blue dots, they could still produce viable offspring, and those offspring would then be hybrids. So that's what we're referring to with the hybrid zone, is that for a period of time, there may still be genetic similarities. The species haven't fully diverged. I would say one example that would fit with this would be gray wolves and domesticated dogs. We know that there are significant phenotype differences. Just put a little teacup chihuahua or a little mini bully next to a great big old wolf, and you'll see some significant phenotype differences. But if the wolf and the little teacup chihuahua were both in their breeding zone or breeding time, they could mate and produce viable offspring. So there are still enough genetic similarities between wolves and domesticated dogs. Even though they're separated and isolated, they could still produce viable offspring, and those viable offspring would be, if we're talking about teacup chihuahuas and wolves, would be unbelievably ferocious, because chihuahuas, they can have an attitude. So we know that there's enough similarities we could get some of these hybrids. Now the big question comes down to, when do we get enough accumulated genetic changes? When will the domesticated dog no longer be able to interbreed with the gray wolf? When will we finally have two very separate and distinct species that can no longer interbreed and produce viable offspring? How much longer will this hybrid zone exist? For some species, it's just a few generations. And for some species, you could be talking millions of years where this could still be happening. It all becomes very individualistic to the species themselves. We also have to keep in the back of our mind that every species is on their own time frame. So if we're dealing with something like a mouse, mice don't often live to see their first birthday. So they have a very different time frame with which they're living in compared to something like a crow. Remember, a crow can live to be 20 years, and they don't even really start to reach reproductive age until 4 or 5 years old. So they're on a very different time frame. And then let's think about an extreme example like a redwood tree. Wow, thousands of years. Talk about a very different time frame. That then changes our understanding of how long a hybrid zone can exist, and time becomes absolutely crucial to understand when we are making our observations. So when we are looking at speciation, a lot of times it's important to focus in on geology. And in this case, one type of speciation that can happen is allopatric speciation. This is where we have a geographic barrier that's separating those populations. It's keeping the gametes separate. And so we don't see interbreeding. We may not see an hybrid zone. And so in our population of pine trees being separated here by the canyon, We know that on either side of the canyon, our species are going to experience different abiotic and biotic factors. That's going to be the selective pressure for changing genes or changing gene expression, developing mutations, all of the things that can lead to speciation. After a period of time, we get two very separate and distinct species. So this example of the salamander, I think, is really nice for helping us understand allopatric speciation, because moving north to south or south to north, California is really, really big. And then we've got some significant geographic barriers there. We've got some large mountains, and salamanders are very, very tiny. So already visually, hopefully you can see how that geographic barrier would keep populations separate, and then also start to understand some of those different abiotic and biotic factors, the temperature differences, the experience of snowfall, the changes of seasonality. I mean, if we're talking San Diego, that doesn't really fluctuate much, doesn't have a winter, doesn't have a summer, it's like always the perfect temperature, versus something like Northern California or in the mountains where you get a lot more extremes. We can see why that would start to put pressure on the different populations, and why we would see allopatric speciation start to occur. Now, with sympatric speciation, we're going to see genetic changes. We're going to see speciation happen within a population that's living in the same geographic location. So some individuals start to experience different abiotic or biotic factors that are driving changes in gene expression, but they're in the same geographic location as the other organisms. And so the reason why I like this example of the pine trees is because trees don't move. So if this population of pine trees happens to be planted or happens to be growing in a section of soil that's more acidic, or is wetter, or has a different composition, or happens to be polluted. Let's say we're dealing with those tailings, the result of mining operations, where within that soil we could have really high levels of mercury or arsenic or other toxic chemicals, but it's in a fairly isolated pocket of soil compared to the surrounding area. We would see that those individuals, if they're going to survive there, they're going to need to change their genes or change their gene expression. So we could see speciation start to happen within the same geographic location. So I think with plants, this makes a lot more logical sense when first learning the concept, as opposed to some of the animals. But I do give you the fish example to help you understand sympatric speciation. We tend to see this a little bit less frequently. So here are several different types of speciation that we see. Again, giving you another way to visualize how speciation would happen over time, what those drivers might be. Notice that we're just focusing on the two big ones here, allopatric and sympatric, but you may go into ecology or other areas where they talk about peripatric and parapatric speciation as well. So our last big concept that we're going to cover with this chapter is phylogeography. And this is super exciting because essentially what we're doing is we're taking phylogenetic trees, which you know is based on morphology and DNA comparison and RNA and protein comparison. We're taking those phylogenetic trees and we're mapping them onto the geographic location. So this first example shows us the different populations of giraffes. And the interesting thing about those is that they are geographically isolated from one another, and we see morphological changes. So our phylogenetic tree is showing the relatedness between these different giraffe populations, and then we've mapped that onto the geographic locations across the continent of Africa. So this is really, really neat because we get lots of scales of information that helps us understand the bigger situation. Giraffes, while beautiful and wonderful and very important to the ecosystem, this isn't necessarily as much of a big impact as, say, something like cicadas in New Zealand, especially if we're trying to plan when the next big cicada population boom is going to happen. So here we have the phylogenetic tree of different cicada species in New Zealand mapped onto their geographic locations. And this next one is really important for bacteria like tuberculosis. And so one of the things that people wanted to do is they wanted to figure out where this highly antibiotic resistant strain of tuberculosis came from. And in order to do that, they needed to sequence the tuberculosis bacterium genomes, look for significant pieces on the genome, and compare that to the populations across the entire planet. So then they were mapping the DNA changes in the bacteria to the populations, and this helped them trace that antibiotic-resistant strain back to a prison in Russia. And then they were able to trace the spread. Now, the reason why this becomes important is imagine if you are a politician or a health official or you work in a hospital. And one of the things you want to know is, am I going to see this antibiotic-resistant strain show up? If yes, then I got to change what I'm going to do. I have to, deal with that population separately. I'm going to have to isolate them. I'm going to have to contact trace all of the people that they may have come in contact with. You have to take action, especially since antibiotic resistant means you can't kill it. So what if in my community of Rockford or Roscoe or Byron or Pecatonica, what if in that community I have individuals that have this type of tuberculosis? How am I going to cope with that? What am I going to do to support them? And how am I going to control this and make sure it doesn't spread beyond my isolated area? Now, hopefully, this helps you to understand phylogeography is exactly what we've been using with the COVID virus, tracking those mutations. Where did they come from? And it's also one of the reasons why it's absolutely crucial that we don't spread the virus and the bacteria. Any virus, any bacteria. Because the more we spread it around to more organisms, the more mutations can happen. Each new body has a different immune system that puts pressure on that species to develop gene expression changes. This better sound like I'm talking about speciation, because that really is what I'm talking about. Our bodies can drive the bacteria, can drive the viruses to go through speciation, to develop changes. Sounds a lot like directional selection. So, we want to know where these changes are happening. We want to track them across the globe in the different populations. It helps us to be more informed. We also want to make sure that we just don't spread these things in the first place. We don't want the changes to accumulate and to occur. So very powerful, but I'm starting to sway into molecular genetics. I'm starting to sway a little bit into evolutionary medicine, which we'll get to. And that's really what happens in the second half of the semester, is everything starts to come together and fit together. And I hope that you're feeling that at this point. So in chapter 11, we deal with macroevolution and extinction. And again, we're focusing on time. This time, we're changing our perspective and we're looking at, first of all, how long has our planet been around? And so it's important to pay attention or touch base with our Earth clock, our clock of how long our planet has been here, when life originated, the evidence of our oldest fossils. And then remember that even the dinosaurs, mammals, and humans are relatively recent on our planet. And then we also have to keep in mind how slowly continental drift happens. So we know that the crust is broken up and the crust is moving, but this movement is so slow. And we think back to things like the ice fish and how long it took for South America and Antarctica to separate. And during that time, that gave the ice fish a chance to be able to develop changes in gene expression. And for any failures in those changes in gene expression for those individuals to die before they had a chance to pass on their genes, and for only the successful ones to pass on the genes to the next generation, such that we get millions of years down the road and the ice fish have this amazing ability to be able to breathe through their skin and have antifreeze in their blood and take advantage of this new niche that was developing as those continents were separating and splitting apart. Remember that the concept of Pangea and continental drift is why we find similar fossils on very different continents and very different geographic locations, and that The slow speed of continental drift is what allows species to stay the same, punctuated equilibrium model, but also gives them the ability to change in response to changes in the environment. So when we look at something like adaptive radiation primarily happens on islands. And the key thing here is that with adaptive radiation, our species on their island were once very closely connected to the species on the mainland. We had two populations that had gene flow flowing back and forth. So if we think back to that hybrid zone arrow that I showed you, the populations were going back and forth and back and forth. We had that gene flow, breeding individuals, moving their genes and interbreeding with different populations. So we had a lot of similar genetic, genetic similarities between the two populations, And so as continental drift is happening and that island is getting separated from the mainland, there comes a point where less and less individuals are able to make that journey back and forth to the original population. So we start to see gene flow decline, and then there's the day, I don't know, is it a Thursday in the year, blah, 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 or whatever. But there's a point where the island population can no longer reach the original population. And so then the differences in abiotic and biotic factors drive speciation events to occur. Now with that adaptive radiation, we have an extra layer here that's a problem. If you're stuck on an island, your population is going to grow very quickly, and very quickly you're going to start to compete with each other. And that competition with each other is going to then drive speciation. It's going to drive individuals. It puts selective pressure on individuals to develop mutations, to take advantage of new resources, to move into a new niche. And so we tend to see with adaptive radiation that competition is a major driver. Now, it isn't always that way, but it is a lot of times. And so if you can start to develop adaptations to take advantage of new niches, that makes you more successful. You're more likely to pass it on to the next generation. And within a certain amount of generations, we now see speciation occur. And some of those phenotypes that we see are pretty extreme. And so we're looking at something like the honey creepers, which I think is a good example of adaptive radiation. We see those extremely curved bills. And that's generation after generation after generation. We see a lot of selective pressure to take that structure that already existed, to eat insects or to crack seeds or whatever you were doing with your beak. We see that selective pressure to change and modify that existing structure to take advantage of a new resource. Now, what you and I see today, if we were to go to Hawaii, is we would see these extreme phenotypes. We would see this very intricate, very tightly coupled relationship between these birds and their resource, such that I can't take the resource away. I can't alter that resource in any way and still have the other species survive. So this can be a challenge. I like to think of it as evolutionarily, you've painted yourself into a corner. You as the species are so finely adapted to this one resource that if anything happens to that one resource, you are going to go extinct. Now, this is the problem that we find with humans, because humans come in and we don't necessarily pay attention to the nuance in the environment. We don't understand all the relationships. We have a certain amount of arrogance and naivete and just we want what we want. And so we go and we get what we want. And later we look back and we go, oh, see, I cut down that tree with the really interesting shaped flower. I didn't think anything about it, but there was this whole species of birds that was their only food. And when I messed with that, I messed with these other species. And I didn't realize that these other species were then connected to the other things in the ecosystem and the other things in the environment. So this can be very concerning because with adaptive radiation, we can see such specialization. We can see intricate relationships that have been formed between the different species in the geographic location. So the other thing that we need to talk about, or at least briefly brush by, is animal evolution. Of course, that's because we're animals. So I wanted to make sure to put this in there so you at least had a little bit of a sense of where we came from and this idea that we are connected back to the rest of life on this planet. So remember our three domains, bacteria, archaea, and eukarya. We are part of the domain eukarya, and we are actually more closely related to fungi than we are to plants, but that kind of makes people a little bit uncomfortable, because if I tell you you're related to a mushroom, well, people look at a mushroom and they go, no, how can mushrooms be related to humans? But it all comes down to the DNA. So where we're going to pick this up is we're going to pick this up at the ancestral protists. So we have protists that then are the ancestral, last most recent common ancestors to everybody in our animal kingdom on the phylogenetic tree. And here we see that this phylogenetic tree focuses on everything except for those of us with a spine, and mammals and animals. And I wanted to put this perspective on things like mollusks and cnidaria and porifera, because we forget that they're with us on the phylogenetic tree, that we are closely related to them, or at least more closely related to them than we are to the other life on this planet. And I also wanted to point out that there's more to the animal phylogenetic tree than just mammals and birds and lizards. But of course, we do tend to like those fish and mammals and birds and all the things with a spine. So let's take a look at our last most recent common ancestor to all animals and protists. These are the caroflagellates. So these are very, 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 very distantly related organisms. Through the Precambrian explosion, we have evidence of multicellularity. We have that evidence of soft-bodied organisms, but it's really hard to get a lot of details because they were soft-bodied organisms. Soft-bodied organisms don't fossilize very well, so I'm sure a lot of the structures have been lost, and we have to extrapolate a lot of conclusions about what these structures were about, how these organisms live, were there predator-prey structures, was there sexual reproduction, how were they doing that, all of the different things that we now take for granted in organisms that we can study either with fossils or living organisms. And we jump ahead to the Cambrian explosion. We had all of that Precambrian speciation, all of those different attempts and twiddles at multicellularity and how to be life on this planet, animal life on this planet. And then we get the Cambrian explosion, driven by changes in the environment. And one of the big important things with the Cambrian explosion is we see modularity, these repeated structures, and we see some structures that start to look more familiar. So there's a lot of diversity in this time frame, and we have a lot of good fossilized evidence of those organisms. This species, we just got some recent evidence and some data about what this species has done and how it's related to life on this planet. And the idea that this is related to leeches. I don't know if you've had any leeches on you, but I'm glad that they don't have all those spiky structures. They're already scary enough, much less if they had any additional structures included. Now, the next step is to start to figure out what those structures might be for. Were those structures for walking? Were they for defending against predators? And that's very fun to think about, all of these fossilized structures on organisms that we're not familiar with, and what all of that might be about. But we have to remember that we are looking at these from our point of view based on our experience with life on this planet. There could have been a lot of other things going on that we just don't know about. But we won't be able to know it because we have our paradigm based on our experiences. They might be doing things that were outside of our paradigm. Crinids, as we see in this picture, were also animals. You can see why they might have been considered to be plants. But again, as we're looking at how do we get humans, how do we get life on this planet as we know and see now, especially animal life, this is one of our ancestors, our crinoids. And of course, the trilobites. And if you remember, the trilobites are really interesting because we've got a lot of morphology. We can see that there's variance, so we can extrapolate that these are all separate species. But because We don't have any living examples of them. We have to extrapolate the relatedness between these different species based on their age, based on radiometric dating, and of course, the law of superposition to help us understand who came before whom. So we've got a lot of potential pictures on how life might have looked as we have that diversification happening during the Cambrian explosion. I wanted to have this map in here because some of you may be avid fossil hunters. And if you go to these places where there's the Burgess shale, you can crack open fossils everywhere. It's really, really easy to find all kinds of different fossils. And what's beautiful about the shale is that it falls apart in these nice little layers, almost like breaking apart loaves of bread, just a bit more solid than a loaf of bread. And here's a picture of what that Burgess shale would look like if you were ever in the mountains and wanted to start looking for some some skeletons. Now, we're touching just briefly on the highlights of animal evolution, and we want to build up to humans and where did humans come from. That's why we're getting this background information. Now, for each of these different groups within the animal kingdom, I want you to focus in on the derived characteristics and then all of those ancestral characteristics. So we start with porifera, those sponges. Then we go to cnidaria, and here's a beautiful example of that with our jellyfish. And then the nematodes. Oh man, there are so many nematodes in the soil. They're so important to soil health and thus the health of the rest of the ecosystem. Then we move to mollusca. And when it comes to our mollusks, remember the eyes and the evolution of the eyes and how the squid eye and the snail eye and even the abalone eye, how those developed. So I know it's easy to write these species off and to say that they're not very sophisticated. And yet, if you were to watch the documentary, My Octopus Teacher, I think that you would be really deeply impressed with the intelligence, and the documentarian catches just some beautiful, amazing footage that nobody had got before about their behavior. This is a very dynamic group that we have often overlooked. So mollusca has some very important ancestral versus derived characteristics, as does Annelida, are very highly segmented organisms. We then move to our arthropods, and then our things like starfish and sea urchins. So that takes us all the way up to the group of species that have a spine. And that, of course, tends to be the group that we are more familiar with. So when we look to our mammals, we see that there is this continual building of what has come before. That's why we pay attention to those derived characteristics, because those derived characteristics end up showing up in us. So, the reason why we skip over the evolution of fish and amphibians and we go right into lizards and birds is because of our Into the Jungle chapters. And I know it seems like we're reading a lot with having both chapter 5 and 6 coupled together, but they really do couple nicely with these two chapters in this unit. So down near the bottom of this phylogenetic tree, if we follow it all the way, we see our crocodiles and birds, closely related to one another. In Into the Jungle chapter 6, you're going to be reading about the dinosaurs and why the dinosaurs went extinct, building our way into the birds. And I want you to have this visual of the KT boundary, because I think that it's helpful to have that in your mind as you are reading the textbook chapter. So you may want to go right now and start reading Into the Jungle chapter 6. You may want to start reading that, maybe get a bit confused, and come back and get this visual aspect to help you understand what's going on and why that textbook chapter is so important. Now, I think in addition to reading the textbook chapter, if you want to procrastinate better, or if you want to do this for an extra credit assignment, there is a beautiful, wonderful PBS Nova episode on the day the dinosaurs died. I think they do a fantastic job of talking about the KT boundary, why that was important in helping put all of the puzzle pieces together to figure out why the dinosaurs went extinct. And you have to remember the dinosaurs have a tremendous amount of diversity. So we had a lot of speciation events. There was time for those speciation events to occur. The environment was consistent for long periods of time. We could see this competition-based, resource-based driving of speciation, and then gone. Most of it just disappears. And that's why we get into the jungle chapter 5, is because we have this situation where for a time mammals and dinosaurs are living side by side. So all of that diversity that we saw leading up to getting a spine and mammals on the phylogenetic tree, all of that was happening at the same time as the dinosaurs are wandering around, and we've got mammals living alongside dinosaurs. And we've got this idea that dinosaurs were laying eggs. So we've got these two really groundbreaking observations that happen, that challenge the existing paradigm, and that people are still struggling with today. And so when we look at those early mammals, they were incredibly tiny. Of course they would be. The dinosaurs dominated. There was so much speciation that you were going to have to be very specialized if you were going to take advantage of a niche and you were going to survive with all of this competition. So here's some visuals to go along with the mammals that existed alongside the dinosaurs. And they were not very dramatic at all. So the big question is, how do we get from mammals that look like this to us? And that has to do with extinction. Now, I know that we tend to think about extinction as being this horrible, awful thing, and it is for the individual. I mean, I'm a Homo sapiens, so if the Homo sapiens go extinct, that's going to be sad. It's going to really stink. I'm not going to like it. But to the rest of the planet, I don't know if the rest of the planet is really going to care. You know, I mean, as soon as we're gone, there's a lot of resources available. If there's resources available, then something new is going to move in and start to take advantage of those resources. So what we see, we look backwards on our planet, is that we see consistency in extinctions. They happen. And it's our values that judge them as being bad. I mean, if the dinosaurs hadn't gone extinct, we may never have gotten to humans. So I think we need to really think about and talk about the concept of extinction from a point of view of what is this organism's role in the ecosystem? What are my values associated with that? And if this organism is not here, what moves in to take its place? So every time we've seen a mass extinction, something has moved in and been able to take advantage of those resources. It's almost a law, but I can't go there because remember, laws are omnipotent. They apply everywhere in the universe. So it seems to be a very consistent rule. We as humans have been deeply altering our planet. We have altered the climate, the water, the ocean circulation. I am a bit bothered by the recorded events in the Atlantic Ocean, that the ocean currents are slowing down significantly. That is disturbing because our ocean currents have such a larger impact on the weather and the climate that we experience on land, not just to mention all of the cycling in those large bodies of water and all the life that still lives there. I mean, every part of this planet is intricately connected. And so when we look across this chapter, we see that both chapters, we see that time is very important. Time for species to be able to change. Time for genes to be selected, to be passed on to the next generation. For unsuccessful attempts to be eliminated and to disappear. Whenever extinction happens, new organisms will move in, but it will take time for them to be able to take advantage of those resources and speciation to occur in response to the extinction. We see these large-scale patterns. We can look back and analyze them, figure out a lot of the whys behind the observations that we're making. And that can help inform us about the present and help inform us about the future. And so when we start to envision, what do we want our planet to look like or to be like in 100 years, 500 years, 1,000 years? One of the things that's interesting, if you ever watch the show Finding Your Roots, is when they can trace people's ancestral connection back 500 years. And stop to think about, did that person think that there was ever going to be a great kid looking back and going, I am genetically connected to you. I see the events of what you have done because you recorded it and other people recorded it. I am looking back on your life. I am connected to you. I am having this other experience. We don't normally think that far into the future. We don't think that it's possible to exist. And somebody 500 years from now may look back on us and say, you did the following things. I am connected to you. Here's where our life is at now. Well, we have that ability. We have that ability to look backwards, to be in the moment, and to look forwards. That's what I love about evolutionary biology. So I hope that these chapters can help you to understand more about our planet, about how mammals came about. Of course, our next unit, we will be focusing in on human evolution. As always, email me if you have any questions.